Hello everyone, this is our second lectures of Mendel's law of inheritance for NEET exam. Now, today we'll talk about the different laws of Mendel's. Like first law, law of dominancy. Law of dominancy, it states when two contrasting alleles for a character came together in an organisms, only one is expressed completely and so visible effect. This allele is called dominant allele, and other allele of the pair which does not express and remain hidden is called recessive allele. Means কি হয় না দুটোর মধ্যে যেটা ডমিন্যান্ট অ্যালিল সেটা এক্সপ্রেস করে এবং রিসেসিভ অ্যালিল কি হয় সাপ্রেস থাকে। নাও ইফ ইউ সি দ্যাট টল প্ল্যান্ট এন্ড ডুয়ার্ফ প্ল্যান্ট এদের মধ্যে যদি আমরা ক্রস করাই তাহলে কি পাই ক্যাপিটাল টি স্মল টি দ্যাট ইজ টল। Tall and dwarf, that is tall is the dominant and dwarf is the recessive one. Tall is the capital T small t. Now law of dominance is deviation of law of dominance is incomplete dominancy বা অসম্পূর্ণ প্রকটতা and co-dominancy বা সহপ্রকটতা. Now we can see that incomplete dominancy. What is incomplete dominancy? In case of incomplete dominancy, you can see when F1 hybrid exhibit a mixture of character of two parents. F1 hybrid এর মধ্যে যদি আমরা মিক্সচার পাই, প্যারেন্টস এর যে ক্যারেক্টার তার মিক্সচার যদি আমরা পাই তাহলে তখন কি হবে ইনকমপ্লিট ডমিনেন্সি। যেমন যদি আমরা একটা রেড ফ্লাওয়ার ক্রস আর হোয়াইট ফ্লাওয়ারের মধ্যে ক্রস করাই তাদের মধ্যে যদি আমরা পিঙ্ক ফ্লাওয়ার পাই তাহলে these are incomplete dominancy. Right? So if F1 hybrids self-crossed, it produce F2 generation. Genotype and phenotypic ratio in case of incomplete dominancy, genotypic and phenotypic ratio both are same. Like you can see that here, capital R, if you put in a Punnett square, then capital R smaller, then we got it that red is to pink is to white, 1 is to 2 is to 1. That is genotypic and phenotypic ratio both are same in case of incomplete dominancy. Now in coming to co-dominancy, here both alleles of a pair express themselves fully in F1 hybrid. Both alleles are expressed together, like in AB blood group, MN blood groups are examples of co-dominancy. Now coming to law of segregation or law of purity of gametes. According to these laws are says alleles are segregated from each other during gamete formations. And law of independent assortment, what it says, alleles not only segregate from each other but also can combine with each other with all possible combination when two pairs of traits are combined in a hybrid, and segregation of one pairs of character is independent of each other pair of characters. That is, the alleles not only segregate, not only separate, they are independently assorted, independently transfer into the gametes. And that's why the practical examples of law of independent assortment is that when you see the family at a family, there is two children, one is it's a two children are different from each other due to this law of independent assortment. Now in case of F1, the phenotype of F1 hybrid resembles both the parents, is it will be one option in incomplete dominancy, law of dominancy, inheritance of one gene, it will be co-dominancy will be the answer. Both parents resembles both the parents, here answer will be co-dominancy. So example of co-dominancy is like red and white, and in mixture one is came, that's the example of co-dominancy. Co-dominancy also phenotypic and genotypic ratio 1 is to 2 is to 1. Now coming to the dihybrid cross of Mendel. Now dihybrid cross, well, you see that yellow round seed and green wrinkle seed. If we cross, then we got this as a F1 generation. Then here we get the checkerboard. These are gametes. Now you can see that the genotype capital R, capital Y. This is round yellow, capital R, capital Y, small y, round yellow. These are numbers and these are probability. 1 by 16 means this is a total number. You can see that there is a checkerboard, we can got 16. So here say total is 1 by 16, 2 by 16, 1 by 16. It's now you can see the capital R, small r, capital Y, capital Y, round yellow. So 2 number 2 by 16. So in that way we are getting. So here we get round yellow. How many round yellow we get? Round yellow. that is 1 2 3 5 4 9. So say round yellow is 9. Round green is round green. This one round green, and this 2. This is 9 is plus 2 plus 1, that is 3. Wrinkle yellow 3, and wrinkle green will be 1. So this way we'll get 9 is to 3 is to 3 is to 1 ratio. Now coming to test cross. If we cross the test cross of dihybrid, we know that test cross. Test cross is the F1 hybrid with homozygous recessive. If we cross, then we call it as a test cross. Now you'll get here, that is, so 1 is to 1 is to 1 is to 1 ratio. So reciprocal cross also give the same result in dihybrid and monohybrid test cross. If we do reciprocal cross in case of dihybrid and monohybrid test cross, then also you get the same result. Deviation of law of independent assortment is linkage. Linkage, we know that when the two genes reside on the same chromosome and try to inherit together, then their transmission pattern is called linkage. Linkage means, suppose there is a two friends, দুজন বন্ধু আছে, দুজন বন্ধু সব জায়গায় একই সঙ্গে যায়, they are linked together. So when two genes reside on the same chromosomes and try to inherit together, then their transmission pattern is called linkage, and these genes are called linked genes. Linkage can be two types. One may be complete linkage, maybe incomplete linkage. Complete linkage, if 0% crossing over, and incomplete linkage, there is a high chance of crossing over is there. That's the major purpose. Now coming to epistasis. Epistasis is a non-allelic interaction. You know that epistasis is a non-allelic interaction. In case of non-allelic interaction, if one gene modifies the expression of another gene at distinct locus, then are called as a epistasis. Any gene that masks the expression of another non-allelic gene is epistatic to that gene. The gene that suppresses is called hypostatic interaction. Now coming to the dominancy versus epistasis difference. Dominancy is a allelic interaction or allelic suppression. Epistasis is a non-allelic interaction. In dominancy, only single gene involved, but in case of epistasis, more than one gene involved. Dominancy, it involves a single pair of allele, involve two pairs of alleles. One allele is dominant and other allele is recessive. One gene is epistatic gene, another gene is hypostatic gene. So these are the major difference between epistasis and dominancy. Now coming to types of epistasis. Types of epistasis is dominant epistasis, duplicate dominant epistasis, recessive epistasis, and duplicate recessive epistasis. Dominant epistasis, what it means, when the dominant allele of one gene masks the effect of either allele of the second gene, it is termed as dominant epistasis. In case of here, if for exam point of view, we have to remember the ratio of dominant epistasis. Dominant epistasis, we know that the 9 is to 3 is to 3 is to 1. These are dihybrid ratio, dihybrid phenotypic ratio. So if we add the first two, then we got 12 is to 3 is to 1. That is dominant epistasis is 12 is to 3 is to 1. Duplicate dominant epistasis, add first three, these three, then we got as a duplicate dominant epistasis. Duplicate dominance is 9 plus 3 plus 3, that is 15 is to 1. That is a duplicate dominant epistasis. Now next is recessive epistasis. In case of recessive epistasis, in a pair of non-allelic gene, one produces its phenotypic effect independent in a dominant state, but another cannot produce phenotypic effect independently. Now in case of recessive, you have to remember the recessive epistasis, the ratio, phenotypic ratio, in case of if you add the from last one, 9 is to 3 is to 3 is to 1, if you add from this, then you get the phenotypic ratio of recessive epistasis. Duplicate recessive epistasis, add last 3, then you will get 9 is to 7. That is a duplicate recessive epistasis. Now coming to the lethal genes. One gene which causes the death of the organism is called lethal gene. So here is the types of lethal genes like recessive lethal, dominant lethals, and conditional lethal genes. Hexokinase gene for glucose metabolism, phenyl phosphofructokinase genes for glucose metabolism is also one of the important examples. Here in mice coat color, the Y allele, the mutation is cause death of the organism, that is lethal. Now coming to dominant epistasis, already we have looked about dominant epistasis. Now we see the ratio. Now monohybrid ratio, monohybrid cross, phenotypic ratio 3 is to 1, genotypic ratio 1 is to 2 is to 1, test cross ratio 1 is to 1, reciprocal test cross ratio it will be 1 is to 1, dihybrid cross, phenotypic ratio 9 is to 3 is to 3 is to 1, genotypic ratio 1 is to 2 is to 2 is to 1 is to 2 is to 2, 4 is to 1 is to 2, 1 is to 2 is to 1, test cross ratio 1 is to 1 is to 1 is to 1. We have already talked about that part. How we will, if you forgot, you can come into that one. 1 is to 2 is to 1, 1 is to 2 is right. Then you will get it this one. Now come into the test cross ratio 1 is to 1 is to 1. Complementary gene ratio 9 is to 7. Dominant epistasis 12 is to 3 is to 1. Recessive epistasis 9 is to 3 is to 4. Duplicate dominant epistasis 15 is to 1. Duplicate recessive epistasis 9 is to 7. Incomplete dominancy 1 is to 2 is to 1. Genotypic ratio 1 is to 2 is to 1. Co-dominancy 1 is to 2 is to 1. Now moving to co-dominancy, monohybrid cross ratio, phenotypic ratio 1 is to 2 is to 1, genotypic ratio 1 is to 2 is to 1. Trihybrid cross ratio 97 is to 9 is to 9 is to 3 is to 3 is to 1. So this will be helpful. Thank you.